that these rare disease networks have been established for many conditions, and we were lucky to be funded in 2019 to establish our MGNET rare disease network. And just from the beginning, it was a great opportunity, right? Because for any rare disease, there really needs to be a concentration of people who are studying this beyond their simple site. There just are not enough numbers of patients that one can study at a single academic site. And so that's allowed us to produce the largest biobank of specimens devoted to myasthenia gravis that's linked to very rigorous measures of how the patient is doing. So that's just really never been done outside a unique clinical trial in the past, that we really have an understanding of, or will have an understanding of, how patients themselves are doing kind of in the real world setting. We're not manipulating their medications in any certain way. We're just observing them for 2 years and taking their cells and serum to study. Now, that, I think will become just a very rich source of information. So I was lucky enough to be part of a clinical trial called the MGTX trial that demonstrated that removal of the thymus benefits patients with myasthenia, at least the ones with earlier onset of disease. But that was a 3-year study where we collected biospecimens, and now we have 10 publications related to that work, each which has led to really fundamental understanding of the condition that could not have been done otherwise. And we're going to reproduce that success with our natural history study. But along the way, we're doing other things. So, for example, my colleague Kevin O'Connor at Yale, who's a critical part of the MGNET group, has just recently published his work on new bioassays to determine what antibodies activate complement and which ones don't. In a nutshell, the reason that's important is we now also have drugs that inhibit complement, and we can ultimately, hopefully, have an assay that says, oh, this is going to work, or it's working effectively in this patient, or even more importantly, this is not going to work in this patient. And so we can really personalize our medication treatment in the future. My colleague Linda Kushner, again, she's part of the MGNET, has identified a protein called survivin that allows cancer cells to survive. But she's identified this in the autoreactive cells, the antibody-producing cells of myasthenia gravis. So those cells are long-lived and they avoid elimination, very much like cancer cells. So she has a therapeutic development program outside the MGNET, but we then looked at tissue from patients that had therapeutic thymectomy. Again, that's a surgical removal of the thymus that treats myasthenia, and did a multiple set of studies. But the important one here is that the amount of survivin positive cells in the thymus predicts treatment response. So we can have an idea not only of the basic pathophysiology, but an ability to potentially, once verified, predict treatment. And then we have another study that looks at the metabolic composition of patients, so the metabolome, at one time point, and we can predict which patients are going to do well 6 months later, again, with prednisone therapy. So, again, we're trying to find things that can not only educate us on treatment and how our treatments work, but also be impactful for patient care. And the one thing that you asked about, what opportunities has this created? It's I'll just say mind-blowing to me, right? There have been physicians, scientists from across the world that want to collaborate with the MGNET, we're working with an Australian group to try to build a physician registry with agreed upon outcome measures. And this is modeled after a very successful MS-based registry, it's called. So it's been in existence for many years, but it's allowed categorization and treatment response to the multiple FDA-approved medications in multiple sclerosis. And so, we'll be able to, I think, develop that further, partially in collaboration with our MGNET. And then there have been other, opportunities with pharmaceutical companies that want to, utilize our unique resources. We still haven't, come to any agreements with that, but it really has been very exciting to, be part of the rare disease network in and of itself. Just attending our meetings, monthly meetings, learning from my colleagues who have, more experience in the rare disease realm. And, before this, I never kind of thought of myself as a rare disease researcher. I thought of myself as a Myasthenia Gravis researcher. But now I also kind of think very differently about the way I do things, and in a kind of more formalized, educated manner, I'd say, just because of my participation with the RDCRN.